It is my pleasure to kick off the lightning talks with today's first presentation, introducing Kate Rosner of METiS. METiS is applying AI and machine learning to revolutionize drug discovery and delivery. Welcoming to the stage, Kate Rosner.
Hi, everybody, and thank you, Trisha and ILP, for hosting us today. My name is Kate Rosner. I'm going to be presenting a quick overview of METIS's capabilities. So, METIS aims to integrate artificial intelligence, machine learning, molecular dynamics, and high-throughput in vivo screening to develop novel genetic medicines, particularly in the pursuit of easing delivery issues. We were founded in 2019 by 3 MIT alumni, and this mission has allowed us to raise over $300 million through our Series C round. Jacob actually this morning gave a really good introduction into why AI is a really good application, or why LNPs are a really good application for AI and machine learning. There's a really large design space when it comes to LNPs. You can design chemically each individual component of the LNP, but you also can optimize the LNP particle as a whole by adjusting the proportions and ratios of the different components within the particle. So that creates a really large combinatorial explosion problem, makes it really difficult to design these LNPs optimally with just traditional experimentation or DOE. So we use AI, machine learning, other computational tools to solve that problem. This all begins with our lipid library. It's over 1 million different lipids, not just ionizable lipids, but it also includes other components of an LNP. This is all in silico, and it was generated de novo by machine learning algorithms. Obviously, we can't synthesize millions of lipids, so we have in silico AI and molecular dynamics filters to allow us to quickly screen through these lipids and other LNP components to identify which would make the optimal LNP for delivering to a target tissue of interest. And then finally, we use artificial intelligence to predict the optimal formulation as well to best tune these LNPs for efficacy to delivering to these difficult to reach tissues. Like most LNP companies, we began with developing liver-targeting LNPs. So we have liver LNPs that have better delivery than Moderna's Lipid 5, which is kind of like the industry gold standard. And this portfolio of liver LNPs that we have actually supports our internal pipeline, which is primarily focused in oncology. So this MTS 105 is one example of our pipeline. It is an liver-targeted LNP that encapsulates mRNA, which encodes for T-cell engaging scFvs. This is for the treatment of hepatocellular carcinoma. In vivo, we've seen significant tumor shrinkage with this therapeutic, and we actually have finally dosed in humans in an investigator-initiated trial that is still ongoing, so that's very exciting. But beyond the liver, we've also developed an extrahepatic targeting LNP portfolio that allows us to target a number of difficult-to-reach tissues with our LNPs. So we have CNS-targeted LNPs that can be administered by intrathecal or ICM administration. In the slide, you'll see we have ICM administration of TD tomato mRNA. RNAscope shows that we're seeing really widespread distribution throughout the brain, including to deep brain regions. We also have lung-targeting LNPs via IV administration. We have seen better delivery and more specific delivery to the lung versus the liver and spleen in comparison to SORT LNPs, which are the basis for RECODE's foundational technology. And finally, our kind of like breakthrough, really unique technology is IV administered skeletal and cardiac muscle targeting LNPs. These give us a lot of opportunity to potentially help patients with diseases with significant unmet need. We're thinking like muscular dystrophies or cardiomyopathies. It's really exciting data. So I know it was a lot to get through in a very short period of time, but I'm open to hearing from any of you. Please come find me at the end of this. I'll be happy to talk more about what our company is doing. Something I wasn't able to get to here is we actually have an AI-empowered mRNA platform as well. And we are always looking for external collaborators on any of the technology that I've shown you today. So thank you all.